Good day, everyone. Thanks for attending this talk. My name is Shahin Nasr, and I'm presenting our study about the impact of amblyopia on the function of stereo and motion-selective clusters in human extrastriate cortex. Before starting, we need to inform you that among the authors, Dr. Bex has financial interest in adaptive sensory technology, and Dr. Hunter has financial interest in luminopia and Ribion. The rest of authors have no commercial relationship to report. As you may know, amblyopia is the #1 cause of vision loss in children and young adults. This developmental disorder is caused by disruption of binocular vision in early stages of life. Amblyopic individuals typically show decreased stereo acuity and impaired global motion perception. However, neuronal deficits underlying these perceptual impairments are still unclear. This is mainly because our access to the brain sites that are involved in stereopsis and motion encoding is generally limited due to their small size compared to the spatial resolution of neuroimaging techniques. To better clarify this problem, this slide shows the organization of stripes in the dorsal part of human V2 based on using cytochrome oxidase in a postmortem histological study. To help you see these stripes better, we have added these dashed lines on top of them. Among them, a subset of the stripes are homologs of monkeys' thick stripes that are involved in motion and stereopsis encoding, while the others that are homologs of thin stripes in monkeys are involved in color perception. But as you can see in humans, it's hard to distinguish these stripes from each other based on their shape. The small size of these stripes also make it difficult to visualize and to distinguish them based on their stimulus selectivity in vivo using fMRI techniques, which have a resolution close to 1 centimeter. Our study was based on 4 experiments, all of them based on using high-resolution fMRI collected in an ultra-high field scanner, which gave us a nominal resolution of 1 millimeter. Using this technique in the past, we have been able to localize motion, color, and stereoselective columns across early visual areas. Here we use this technique in amblyopes and control to localize these stripes and sites and to compare their size and response properties to different stimulus conditions. To this day, 27 individuals have participated in our study. Among them, 7 were strabismic and 5 were anisometropic, but they all showed impaired stereo acuity based on RANDOT test. Controls were age matched to amblyopes. They showed normal or corrected to normal visual acuity in both eyes and significantly better stereo acuity compared to amblyopes. During experiment 1, we localized the motion-selective areas in our subject based on the response evoked by radially moving versus stationary concentric rings. Experiment was block design. In 50% of blocks, stimuli were moving, and in the other 50%, they were stationary. By contrasting the response to these stimuli, we were able to localize the motion-selective sites. During experiment 2, we localized the stereoselective areas in our subjects based on the response evoked by 3D versus 2D stimuli generated by random dot stereograms. Subjects saw the stimuli through red and green filters. For further details, you can go to this paper. During experiment 3, we localized the color selective sites in our subject based on the response evoked by color varying versus luminance varying stimuli. This test was an important control because we did not expect to see any impairment in color perception in our amblyopes. Also, color selective sites could be used to better define the border of motion and stereoselective sites, as we have shown in our previous publications. Among our subjects, 9 controls and 9 amblyopes participated in experiment 4, during which we measured the coherency sensitivity of the response evoked within the motion selective sites. Here, stimuli were random dots Their size changed with eccentricity to compensate for the cortical magnification factor. In different blocks, we changed the coherency of motion between 30% to 95%. During all experiments, we asked our subjects to fixate on a centrally presented object and to do a dummy task on it that was irrelevant to the background stimuli. The sequence of experiments were also counterbalanced across subjects. Here you can see the location of those cortical sites that show a significantly stronger response to moving compared to a stationary stimuli in one control subject. Red to yellow colors indicate the p-values of 10 power minus 2 to 10 power minus 6. Border of visual areas indicated by black dashed lines were defined retinotopically based on separate scans and stimuli. In macro level, you can see a motion-selective area MT, an extension of motion-selective activity toward area V3A. In finer scale, you can see activity in area V2 in stripy patterns, V3, and also V4. Here you can see the stereoselective sites in the same subjects. As you can see, the stripy patterns are present in V2, V3, V4, and also a strong activity in area V3A and lateral occipital complex. And here you can see the color selective activity in the same subject that starts from V1 and then extends to V2, V3, V4, and also to area V3A, and almost no color selective activity in V3A. This slide shows the interdigitation of motion, stereo, and color selective sites in the same individual. Red, green, and blue colors show the location of motion, stereo, and color selective sites, as you saw in the previous slide. Here you can see more examples of this phenomenon in 12 different hemispheres. Here you can see the motion selective activity in one amblyopic subject with no measurable stereo acuity. Similar to the control subject that I showed, you can see area MT, an extension of activity toward area V3A. And in the finer scale, you can see a stripy activity in area V2, V3, and also V4. Here you can see the response evoked by 3D versus 2D stimuli, and as you can see, there is almost no activity. This is consistent with the fact that this individual was almost stereo blind. And here you see the activity evoked by color versus luminance varying stimuli in the same individual. Again, similar to the control subject that I showed, you see V1, V2 stripy activity, V3, V4, V8, and almost no activity in area V3A. Here you can see the interdigitation of these sites. Again, as in control, color and motion selective sites are interdigitated, but here you see almost no stereo selective site. You can see the same phenomenon here in 12 different hemispheres. In almost all tested individuals, stereoselective sites are rare, but interdigitated color and motion selective sites are easily detectable. So the take-home message for experiments 1 to 3 is that amblyopia has a strong impact on the evoked stereoselective response. This is consistent with the studies in animals that show the decrease in the number of stereoselective neurons due to amblyopia. But it appears that color and motion selective sites are detectable in amblyopes as in control. As the next step, to reveal the reason underlying impaired global motion encoding in amblyopes, we measured sensitivity of the response in motion selective sites. Here you can see the result. Red represents the activity measured in controls, and cyan represents the activity measured in amblyopes. All activities are normalized relative to the activity evoked by the stationary stimuli. Here you can see that in controls, coherency sensitivity was mostly limited to the central V2 and V3. Also, the decrease in the level of motion selectivity in amblyopes compared to controls was also limited to the central portion of V2 and V3. In the peripheral parts, this effect was either opposite or limited to low coherency levels, not high coherency levels. Here you see the activity measured in area V3A. Since the foveal representation in V3A is very small, we only show the activity measured in motion-selective sites detected in the whole V3A. Similar to V3, here you can also see a significant between group difference due to a decrease in the level of motion selectivity in amblyopes compared to controls in presence of noise. So it appears that in presence of noise, motion selectivity becomes weaker in amblyopes compared to controls. This between groups difference appears to be stronger in those sites that represent central compared to peripheral visual fields. So in conclusion, we showed that high-resolution fMRI can be used to reveal the developmental disorder associated with amblyopia. We also showed that these disorders affect the mesoscale functional organization and the response properties of extrastriate cortex. Here we want to thank our colleagues and sponsors for their helps and support, and without that these experiments could not happen. And also we want to thank you for your attention. Thank you.